Good evening, everyone, and thank you for staying on for the last presentation, which is now between you and the cocktail hour that is coming up. So really appreciate you staying over. I'm here to give you a little bit of a glimpse on 4DMT, which is 4D Molecular Therapeutics. We are harnessing the power of directed evolution for targeted genetic medicines. So what is 4DMT? We are unlocking the power of genetic medicines for millions of patients. We are Emeryville-based in California, Bay Area company, listed on NASDAQ, so it's a public stage, clinical stage public biotech company. About 140 employees. We have fully owned GMP and R&D capabilities in our Emeryville facilities. As I mentioned, our platform is directed evolution. I'll give you a little bit more glimpse in a slide. We start with 1 billion synthetic AAV sequences. We are generating novel capsids and customize those for each of the therapeutic areas that we are in. These capsids then evolve into vectors with sort of interesting payloads that we have. The most important thing is, unlike many of our peers in the next generation AAV therapeutics, we are clinically validated across 3 therapeutic areas, which are shown here: the eye, which is ophthalmology, the lungs, pulmonology, and the heart through our Fabry disease cardiomyopathy program. We have 5 clinical candidates for 7 large patient populations, and we are on our way to become a fully integrated genetic medicines company. Now, some of you may be aware of this Nobel Prize winning technology, which was discovered by Frances Arnold, and she won the Nobel Prize. Our platform originally came from a lab in UC Berkeley, and we then industrialized it for the clinical stage. And this is basically leveraging the power of directed evolution. Our goal in this platform solution is to first take those synthetic libraries that we have, almost 1 billion sequences there. We then develop a targeted vector profile, which is our step 2, which is where we define what is the tissue we want to go after, what is the route of administration, and what is the dose that we need to. Once we have defined the target vector profile, we administer our library of capsids to non-human primates. This is the closest to translation from a human angle. And then we have developed from there 3 clinically validated capsids. Our lead vectors are R100, C102, A101, and I will talk a little bit more about those. Our goal is to successfully overcome, and we have demonstrated that in the clinic, the shortcomings of conventional AAV vectors. So as you are aware of it, and this has been a topic of discussion across many forums, limited delivery with conventional AAV vectors, limited transduction, increased inflammation toxicity, as well as susceptibility to neutralizing antibodies. And we have successfully overcome that across 3 different vectors using 3 different routes of administration. This leads to our pipeline, which is a large sustainable pipeline across, as you see on the left to right, R100 is our intravitreal vector that is used for most of the sort of retinal related eye diseases. And we have wet AMD, DME, XLRP, sclerodermia, geographic atrophy, and then we have multiple other targets that we are working on with our recently announced partnership on Astellas. The beauty of this platform is, apart from that partnership with Astellas, most of our assets are our wholly owned assets. On to A101 for pulmonology, we are in the cystic fibrosis lung disease, and I will walk you through some of our lead programs with some interesting data that we have announced to date. Cardiology is 4D110, which is the lowest in the maroon, which is for Fabry disease and cardiomyopathy. So as you can see, broad pipeline, rapidly moving towards phase 2 and beyond. We hope to be in phase 3 by the end of 2024 and across multiple large disease populations. We have fully integrated capabilities in-house in Emeryville, ranging from product design, where we design our particular capsids, preclinical R&D, CMC with all the analytical QA/QC processes, as well as clinical regulatory capabilities for us to be able to take this forward towards regulatory approval. So let's walk through in brief 2 of our lead programs, and I'll give you a little bit more on it. 4D-150, which is for wet AMD and DME, pretty large scale population diseases, high unmet medical need. There is the VEGF treatments that are currently in the market, but the treatment burden is clearly an area of unmet need given the high number of doses that are required, as well as potential adherence challenges that are there in regards to patient needs. It's a broad epidemiology. Most of you are probably aware, but it's a 12 billion sort of a market that you operate in, largely dominated by the anti-VEGF therapies. Our product design, which combines a dual transgene, so we are able to fit that into an AAV vector successfully, and it has 4 different targets: VEGF A, B, C, as well as the placental growth factor. And we believe that we are the only therapy that is targeting both the VEGF C pathways as well as the conventional A and B pathways. Ubiquitous promoter, and we have a sort of a customized version of the aflibercept transgene as a payload. We are ongoing phase 2 clinical trial right now, and we will be sort of proud to present data on the phase 2 early next year. So as a glimpse to the clinical trial design, we have completed the dose exploration stage, which you can see from the doses 6E9, 1E10, 3E10. These are relatively low doses, which is one of our goals: highest efficacy at the lowest possible dose. We've then moved on to the dose expansion phase 2, which compares 3E10, 1E10, and the aflibercept arm, and we will be reporting data on that in early 2024, as I said. But I'll walk you through the dose. For those of you not familiar with the program, the data we saw in phase 1, which gives us conviction that this will be a highly successful product. Bear in mind, as you look at the dose exploration and dose expansion cohorts, these are the patients with the highest anti-VEGF needs. So they are greater than 12 injections in the prior 12 months. And then we've started now a new cohort, which is the population extension arm, which you see on the right-hand side for the lower VEGF patient who are 1 to 6 injections in the last 12 months. We believe that is more reflective of the typical phase 3 population, and that's why that population extension cohort which we have just started enrolling will be quite useful to us as we plan for a potential phase 3. So what is the data, let me just go through, that we saw in the dose exploration stage? Just a reminder, 3E10, 6E9, and 1E3 in 3 dose levels. And these were the top 15% of highest severity need patients. No toxicity, so we're very proud of the safety that we sort of shown here. No grade 1 inflammation, no hypotony, which was sort of consuming some of the other programs. At the 3E10 level, we have 100% injection free in 36 weeks. If you drill down further with greater follow-up, 75% injection free for 52 and 80 weeks, so almost a year and a half and still ongoing, where the patients are injection free, which is pretty convincing to us that this would be quite a viable modality. On the lower doses as well, we saw 71% reduction on the anti-VEGF injection frequency. As I said, we are now building on this data set of 15 patients with another 50 patients that we will report on in our phase 2 PRISM in Q1 next year. Moving on to the cystic fibrosis program. So this is another big program for us, and we have an update coming at the North American Cystic Fibrosis Conference in November. But again, just to remind, it's a pretty high unmet medical need. You have the modulators there, but then there is also the percentage of patients who are modulator ineligible. We think that's roughly around 10 to 15% who have no treatment modality right now. So our approach here is to design a vector which has a truncated CFTR gene. That's the gene that is required to treat this population, fit into our capsid, which is the A101 capsid here. It's an aerosol version. So unlike the previous one, which was intravitreal, this is an aerosol version that we have. and the promoter is ubiquitous. We've completed our phase 1 dose exploration, and we are now moving forward likely after our second cohort on the dose exploration to the dose expansion phase. This is the dose exploration at 1E15 cohort 1, on which we have already announced data earlier in the summer. And on cohort 2, we will be announcing for 3 to 6 patients in November coming up. It's an aerosol version of the vector. And when you look at the data that we have seen, it's creating quite an interest. So first and foremost, the CFTR expression, which you see in the middle part, 92 to 100%, almost supraphysiological. So that's sort of an expression with an aerosol version of the vector, not yet seen so far. And we have seen evidence of the expression going across all the airways as well as all the cell types within the lung, ciliated, basal, and the goblet cells. In terms of safety, it's well tolerated, no post-dosing 4D710, which is our vector-related AEs or DLTs or SAEs that have been reported so far. These are the patients which are modulator ineligible in this population. Again, the severest of severe patients who have no treatment right now. In terms of, if you look at other sort of parameters and markers on the FEV level, for the 3 patients in this cohort, they had already high baseline FEV levels, but still for one we showed improvement and the other 2 were stable as we continue to report on the 9 to 12 months follow-up. And then there is meaningfully clinical improvements in terms of the quality of life metric that you see across all 3 participants, 6 to 22 points, which is quite relevant and is attracting quite a good amount of attention. So in summary, when you look at, we have 4DMT is at a very critical stage and inflection point. We have multiple catalysts coming up in sort of later half or the last quarter of this year, as well as going into first half of 2024. Across all our vectors, 4D150, 4D175, which I didn't explain because that's a program that we will provide an update related to geographic atrophy, and then 4D710 in terms of lung. 4DMT is pretty well funded, $330 million as of the last quarter. So we have dry powder to run all the way into first half of 2026, which is a pretty big thing given the current macro environment in terms of funding. It's a very seasoned and experienced leadership team. David Kern, our CEO and co-founder, comes from UC Berkeley, from where the technology was originally licensed. And then we have Fred Kamal leading our manufacturing CMC, building all of that in-house. And as you can see, we have other leaders here who are all pretty much experienced in this modality, as well as a very experienced board of directors. I think that's all I have. Thank you for your time and patience, and hope you have a lovely evening today. Thank you.